Thanks, Udi, for the introduction. I made a journey of sorts, starting from physics and trying to get into kind of ecology and more sort of a little bit more applied ecology, let's say, not quite applied, but somewhat less theoretical stuff. But still, I'm very much working on theoretical work and modeling, as you will see. And I just wanted to kind of present some recent work. And given that I am, coming from, well, currently in this sort of agriculture university, these kind of questions of how to look at ecological systems, but in sort of agricultural systems, is something I'm getting a bit more interested in, and brings me kind of to ask different questions. And so it would be interesting for me also to potentially hear some of your perspectives. And so I'll start to try to share a screen here. Let's see. Let's see if it work. And yep, okay, I think this is working. Yeah, and so as was mentioned, my talk is called Nutrient Dynamics in Green Brown Food Webs. And especially given that this is Zoom and all that stuff, please do feel free to interrupt if I'm going too fast. If I am not clear, please feel free to ask, or just say Yuval, slow down. And also, I'm a bit I have a bit of a cold, so I hope it doesn't bother anyone too much in the sense of how clearly I'm speaking. We'll see how it goes. Yeah, so let's start. And so, maybe just from the title itself, there's kind of two parts here in a sense, nutrient dynamics and food webs. And one thing I kind of want to get at quite from the start is whether these are kind of two different topics or not. And this is just two sort of cartoons I found just, Googling it and seeing what comes up. But I think it's actually quite representative. When people think about nutrient dynamics, as you can see here, they're really thinking about, in this case, the soil. It could be also in the oceans. It's the same idea, but they're thinking about sort of recycling processes, also physical processes that happen with kind of inorganic nutrients, all kind of processes like that. Things to do with decomposers, and also to some degree things to do with plants, or generally primary producers, and maybe a little bit of animals, but not much usually. And certainly the focus is more on the soil, on the substrate. On the other hand, when people talk about food webs, it's actually usually very different perspective. It's kind of can be seen here pretty well. There's a lot of this thinking about sort of different what's called trophic levels. So, primary producers, primary consumers, secondary consumers, and so on, as we go up and above, having different sort of plants and animals, their interactions, and so forth. And so it really, the focus is, generally almost never on the substrate, to be sure. Typically, it's also what's called sort of the green food web, as in the food web where the energy and nutrients come from plants, basically. As opposed to kind of relating to decomposers and detritus and things like that. And also the people who are doing this are typically very different people. Nutrient dynamics, at least in ecology, is often done in a sort of ecosystem ecology perspective, looking at the sort of maybe global view of things, or certainly more kind of fluxes and stuff like that. Whereas food webs is done more by, say, community ecology, looking at species, biodiversity, things like that. And to a large extent, these kind of two paradigms don't really talk to each other. And so in a sense, this is kind of, there's a story of how I ended up here. That doesn't matter. But point being, I am finding myself a bit in the middle of these two realms and trying to make sense of things. And that's what I will try to do a little bit today. And so what I wonder perhaps is whether this might be the food web that is relevant to you, or to me, in the sense of being, say, for me, being in an agriculture university, part of what people are interested in, what happens in a crop field, in a field of wheat, for instance. So you might have the soil and the plants growing on it. There might be some detritivores that are perhaps quite relevant in amending the soil properties. You might have herbivores, pests, often that eat the plants, and you're not very happy with them. And there might be some other animals around doing other stuff. And so I'm saying this, because perhaps this might show that these kind of questions I'm going to ask are actually quite potentially relevant to kind of agricultural context, which I do believe is also relevant to the Wakani Institute, as is to my own university. But and also just to give a concrete example, maybe that helps think of what kind of specific systems these kind of ideas of kind of connecting nutrient dynamics and food web properties, where that could be relevant. And so what I want to ask today is basically, how does it work, if you will? So for instance, what kind of, how are the stocks, how are the sort of pools of nutrients distributed, and how are the fluxes looking like? And also, how does it respond to perturbations? And I will focus here on nutrient input. So what happens when you change the nutrient inputs, which is of course something we do every day when we fertilize, crop fields, for instance. And so when you're now starting to think, oh, well, how am I going about understanding this system? How, and also for myself, how am I going to model it? So one view that is quite often used is this, what I would call a plant-centric view. So in the middle of it is some plants. They, of course, take up say, nutrients from the soil. They might have some pests we don't like, and that's kind of but everything is kind of centered on the plants. This definitely, I would assume in agriculture settings, I would assume this is quite a common view, although clearly not the only one. In sort of more ecological settings, you might have what I would call a food chain view. So you kind of focus on the sort of trophic levels of how the energy flows from the bottom up to the plants, up to the herbivores, and up to the predators, and so forth. But you could also have about, well, at least we might not want to forget about the so-called brown food web. So that is what happens with all the detritivores and animals that eat dead material and consume dead material and things like that. And they are themselves potentially consumed by predators. So this is kind of the, sort of closes some form of loop. And that is also a lot of what I'm interested in. So how does this kind of two pathways, this here on the left, the green pathway of plants being eaten by herbivores and so forth, and the brown pathway, sort of animals, consumers eating dead material and things like that, how does that relate? So how does these two pathways affect each other, and how does they impact the system as a whole? And so the way I do this in practice is kind of look at it via using a model. In this case, what I would consider a sort of a relatively simple model, not super simple to be sure, but not too bad, with 6 compartments. And this is the flows, and just to make things maybe a bit more kind of relating back to my cartoons. So here we have, for instance, the plants. They're being eaten by herbivores, which are eaten by predators. These plants take up inorganic nutrients here in N. These inorganic nutrients are themselves sort of come up from organic nutrients in the substrate, that is, for instance, soil organic material, things like that in S, which is itself also consumed by detritivores and that are eating the predators, sort of closing a sort of a funky loop. And finally, I realized that I don't have it here, but kind of a major issue as well is recycling of the material, in particular plants, which would mean an arrow from P to S. I apologize, that should have been here in the cartoon. Sorry about that. But yeah, but this is the kind of the system I have in mind. Just to sort of briefly say, there are sort of equations to this. I'm happy to talk about this later if someone is interested. But for now, I think it's often more useful to just focus on kind of more sort of practical things. So, but if that comes up, I'm very happy to discuss that as well. But yeah, so this is kind of the model as I conceive it. And the idea here is also that to some extent, this really hopefully encapsulates everything. What I mean by that is that, for instance, if I track nitrogen as an example, perhaps I'm interested in nitrogen, and I could ask, how is the nitrogen distributed throughout the ecosystem? Then to a large extent, I am covering everything in the sense of things that kind of rapidly change, at least. Certainly there's nitrogen being accumulated elsewhere and sort of slower timescales, but to a large extent, this really should cover all the sort of nutrient dynamics within the system. And so now comes the question of, okay, I have a model, but I want to, get it to make any sense. I need to parameterize it. I need to understand, how are the stocks and fluxes? How do they look like? And so what turns out to be not so hard is to get data on some things, in particular primary producers and their kind of immediate interactions. For example, a very nice paper by Serbian in 1999 AMNAT shows for a large range of different ecosystem types, various properties, including net primary production, turnover rate, and there's a whole lot of things. But they are mainly focused on primary producers, such as plants or phytoplankton, and their production, their herbivory to how much of them gets eaten by herbivores, how much of them gets recycled into detritus, and so forth. But not so much what happens in the food web above, and not so much what happens in the sort of inorganic nutrients like nitrogen. But nonetheless, it's quite a very good source that I found. On the other hand, as I said, one of the biggest problems I ran into is trying to understand the kind of higher levels of the food webs. And honestly, I was a bit surprised to find out that, to a large extent, there, to my knowledge, there's basically never has anyone tried to do this in a kind of sort of a consistent way, like to really try to understand how these things look like in the sort of higher trophic levels. But what I found very useful is this sort of a software suite called EcoPath. This is a nice logo that they have. And this is mainly what's used for fisheries, apparently. And I guess because fisheries means a lot of money and a lot of people put a lot of effort into this. And in practice, you can get pretty good parameterization. You can get pretty good estimates on various food web properties in aquatic ecosystems, in particular marine ecosystems. But for terrestrial systems, things are actually quite difficult. Nonetheless, after a lot of work of trying to add up all the data, here's this kind of a short overview of what that means. So from these two papers that I said around, they include almost 400 papers on primary producers and so on. I could have found many more than 50 EcoPath models, but kind of to be relevant and so on, about 50 EcoPath models, many aquatic systems, 27 systems to accompany that are terrestrial food web properties that are very, like I had to find here and there some things just to get something. A bunch of studies on nitrogen stocks and some studies on nitrogen conversion efficiency ratios. And so basically quite a lot of papers to get some estimates on a whole bunch of stuff. Ended up being quite a data synthesis project. And I coalesced these into 6 ecosystem types. So forest, grassland, what I call brackish, which is basically marsh and mangrove ecosystem, seagrass, meadows, lakes, and oceans. And the idea here is that these are kind of representative of a wide range of ecosystems. And one can argue a bit of a gradient of sorts of of ecosystems between forests and oceans. And so what do we see here? So this is the first figure, which basically when I started this kind of question, I thought the data here would just be available, and it turned out it wasn't. So this is a month of work just to collect this kind of data. But let's see what we have here. So we have in each of 6 different ecosystems that I mentioned, from forest to oceans, in different colors. And in each one, there's these kind of 6 circles representing the stocks. And as you see here, the kind of the dynamics within the system, right? So for instance, P here, this is the primary producers, this is the herbivores, this is the predators, this is organic material, this is inorganic nutrients, and so forth. This is all for nitrogen and grams of nitrogen per square meter. And the fluxes are the arrows between, and they're in scales of years. And sorry, we can see a bunch of things here. Some of them are a bit more well-known than others. For example, just certainly in terrestrial systems, in forest most obviously, but also in grassland, you can see a kind of a very bottom-heavy system, what it's called. You see here and here the stalks at the bottom are much, much higher than the ones at the top. The top is very low. Predators is very small stalks, biomass essentially, especially when you compare it to, say, the ocean, which is kind of the opposite. You see the circles are not the same size to be sure, but they're quite equal size. And this is what people talk about, having this kind of inverted pyramid. That is where supposedly the biomass of phytoplankton is smaller than the biomass of tuna fish and whales. That's kind of an idea. And this is not quite true, but true to an extent, certainly. So you could see some sort of well-known properties here, but you can also see some things that I guess are not as obvious. Yeah, and so to me, I guess what was most striking is that if I look overall, all these kind of 6 sort of graphs are quite quite symmetric, right to left. That is, the size of the stocks here is relatively close to the size of the stock here. The size of the stock here is relatively close to the stock here, and so forth. And this is kind of true to all of these systems, despite the other differences. And I can show this more sort of concretely. So here I'm comparing the organic nutrients, sort of soil organic matter, if you will, versus the primary producers. So these are the two kind of pillars of the system, if you will. The source of primary producers on the one hand, and the other hand, the organic nutrients that, of course, come mainly from the primary producers, but nonetheless act in a somewhat similar way. And here we have the detritivore biomass versus the herbivore biomass, which in turn consume these two things. Right? The detritivores consume the organic nutrients in soil organic matter form, and the herbivores consume the primary producers directly. And the main point I want to say is that you see here the different ecosystem types, is that both of them seem to fall roughly on the one-to-one line to a reasonable extent. And so we have already this kind of nice correlations between those two things. But what turned out to be a bit more surprising, perhaps, is we tried to then see how these kind of food web compartments, sort of how do they behave, if you will, in an aggregated form. That is, if you look, for example, at all the consumers, all the predators together versus all the herbivores together, how do they relate to each other? And so here I'm looking at two different properties. At the top is consumption coefficients. So basically trying to parameterize how these how these animals, or in this case also plants, how they sort of make use of the nutrients that they consume. And here is a mortality rate, so just kind of natural death, if you will. And the main point I want to make is that if you look in particular at the middle column, which is comparing detritivores to herbivores, again, it's very much on a one-to-one line, and you can see this in the R values of the correlation of the Pearson correlation coefficients, that they're very much correlated, in particular compared to some others, like the predators versus herbivores, for instance, and also to a large extent to primary producers versus herbivores. Here, they're actually quite correlated as well, but not so much when you look at mortality rates. And so this is a lot of data, I understand. The main point I want to make is that it's not only sort of the stocks of the sort of a green, brown. So here, going back, here, what I tried to show is that if you look at the stocks of different compartments, we kind of see a correlation between the green sort of channel and the brown channel, the herbivores versus the detritivores. But what I'm trying to say here is that it's not just the stock, it's really also kind of the overall dynamics, the behavior, if you will, the functional role really of these herbivores versus the detritivores. And so all of this combined seems to suggest that there's some form of essential coordination, if you will, between those the two channels. Somehow, in effect, the way the ecosystem behaves, like uses up living material, primary producer material, is the same as using detritivore material. And I don't know why that is, but that certainly seems to be the case. And so that's one thing I found quite interesting. And then another thing quite different entirely is asking, well, how does these ecosystems respond to change? And you can think of different changes, but different sort of perturbations. But one natural question would be, well, how does it respond to nutrient inputs? This is relevant both because, looking at natural ecosystems where a lot more nutrients are coming into the system for various reasons, whether it's kind of semi-natural forests that are getting extra nutrient inputs or nutrient runoff, or and so on and so on. And again, and of course, in agriculture systems, this is very clearly the case where you're putting fertilizers of different sorts and you're impacting sort of the ecosystem within the agricultural field. And so I wanted to ask here, okay, so how does this, how do ecosystems respond to nutrient enrichment? And so here, using the model, I basically, and the kind of parameterization, I tried to look at for different ecosystem types, so the forest, grassland, brackish, seagrass, lakes, and oceans, and kind of from left to right, how do they respond to nutrient enrichment? Basically, I double the amount of nutrient input, either by adding only inorganic nutrients, which is the full boxes here at the top, or by adding organic, which is showed here by the hollow. And so in the black lines, we see the median values, and sort of the boxes, the 95%, sort of the 5 and 95 percentiles. Now, what I'm showing here is a ratio. So I'm looking how much, for instance, does primary stocks, or how much, for instance, with trees in the forest, how much is their biomass larger following nutrient enrichment, as opposed to the baseline values. And one thing I want to point out straight ahead is that, for example, in some cases, you can see values that are clearly higher than 2. Again, we're doubling the nutrient input. You might have the sort of baseline assumption that the value would be 2. That is, you're doubling input, so you're doubling, going to double whatever else it is. And sometimes that's the case, but it's definitely not obviously the case for. And you can see that there's already here quite large differences between inorganic additions and organic additions. And so now let's look at kind of, oh, this is a lot of data again, but let's looking at all at once here for primary producers, for herbivores, for nutritivores, for predators, for inorganic nitrogen, and for organic nitrogen. And so there's, again, a lot of things one can say about this, but a few takeaways from this kind of look at things. That first of all, if you look at predators versus primary producers, for instance, you see predators are actually much more sensitive to this. For instance, in grasslands, you get more than a tripling of their biomass, of their nutrients, if you will, which is kind of the same, of their stocks, and which is not at all the same for the primary producers. The reason this is a bit surprising, if you will, is because predators are far away in the trophic chain. They're not directly impacted by some nutrient additions, whereas you would think primary producers would be. And so that seems kind of a bit non-trivial. And also the herbivores and nutritivores are also quite strongly impacted in a less consistent way. What you can see here is that herbivores would kind of gain a lot from inorganic additions, but not so much from organic additions, and vice versa for the nutritivores. And the reason is basically that the different additions of nutrients going into different compartments and sort of increasing biomass more of one or the other directly, while at the same time there are predators that are eating both the herbivores and the nutritivores, and therefore you get this kind of switch, if you will. But in some cases, you can see that herbivores are going to actually decrease their size due to nutrient enrichment, which is perhaps a bit surprising. Yeah, and so that is kind of one way to look at the response to nutrient enrichment. And another sort of question you can ask, which I think can be interesting, for example, in the context of an agricultural setting where you sort of are putting in a different nutrient and want to see what the effect is on primary producers, is to look at the net primary productivity. And what I mean by that is a flux of primary productivity left after herbivory. So how much biomass is gained by plants after when we take into account the fact that some of it has been eaten by herbivores. And so here I'm just going to look at kind of two generic systems, if you will, one aquatic and one terrestrial, just for the kind of simplicity. And what I'm looking here is this sort of parameter spaces of two things. Subsidy here, sorry, I should have explained that subsidy is simply the term in ecology for nutrient additions. That's really what that is. So two properties of these nutrient additions of subsidies. On the y-axis, we have in log scale the strength, so the overall flux of nutrients coming in. And then the x-axis is what I call the quality, which is basically from left to right, inorganic to organic. So here at 0 on the left, it's completely inorganic additions, and here on the right at 1, it is completely organic additions. And in the colors, we see just how much productivity we have. I use here this normalized productivity just for clarity. Basically, this is how much net primary productivity there is over how much subsidy strength there is. So if you will, it's something like the nutrient use efficiency, right? If I put in a kilogram of nitrogen, how much biomass in nitrogen do I get back? So that's the kind of what it's showing here. And in gray is just to kind of clarify, this is where within these gray areas is simply where the whole ecosystem is extant. Nothing dies. You have herbivores, you have predators, and so on. And the reason I'm saying this is because to a large extent, you can assume that what happens within this area is the more biologically relevant. You would generally assume that it's not very common to have everything die off. Certainly some species can die off, but if everything dies off, that usually is not very common. So it gives just an indication of what part of the parameter spaces you can kind of get an idea from. And one main thing I want to show here in this specific slide is simply that if we go from left to right, if we switch from inorganic to organic additions, then we can we can ask, how does just the switching, we're not changing amounts, we're just changing, kind of just switching the type of addition, nutrient additions. How does that affect this kind of net primary productivity? Primary productivity after herbivory is taken out. And what we show here, which is somewhat representative, is that in aquatic systems, generally speaking, you as you switch to organic, the amount of biomass of, the amount of productivity will always go down. But this is not at all the case here in terrestrial systems. When in some cases you actually can have the net primary productivity going up. And the reason for that is that when you're putting nutrients into organic, in organic form, of course you're going to get some nutrients that are eventually going to be taken up by plants, but you're also feeding the detritivores, they are feeding predators, and these predators eat herbivores. So if herbivory pressure is strong, then you might get a situation where you're actually benefiting the system by kind of creating a loop where you're feeding essentially predators that are eating the herbivores, and therefore plants can grow without herbivory effects. And this can be seen here in a bit of maybe a clearer way. So I'm using here sort of this made up measure on the y-axis, PSOS, productivity, sensitivity to organic. Basically I'm asking how much productivity changes when you switch to inorganic to organic. A positive value of this measure simply says that when we switch to organic, we're going to have more productivity in plants, and a negative says we're going to have less. And here we show in blue the sort of aquatic system, and in green the terrestrial system, and in dashed lines show systems without recycling of nutrients, and in solid lines with recycling of nutrients. And so there's two main things you can see here. Oh, actually three, I would say. First of all, when you add more nutrients, then the value tends to be lower. What that means is that the benefit of going, if I'm going to assume now that we want more net primary productivity, we want more, we want more yield from our plants. And so in that respect, if we're in a regime that has higher nutrient inputs, the benefit of switching to organic is less strong. That's one thing you can see, that these lines are going down as we go to the right. The second thing we can see is that we can expect in terrestrial systems in green a generally positive effect of having organic nutrients, whereas we're going to expect a more negative effect in aquatic systems. And finally, the difference between the recycling and non-recycling is kind of interesting. What it basically means is that in systems that have less recycling, we're going to expect a more positive effect. In systems, most obviously systems that don't really have much recycling is really relevant for agricultural systems, where recycling effect is relatively minimal for various reasons, really. But the bottom line is that in agricultural systems, this kind of positive effect of putting organic nutrients should be stronger, more so than in natural systems. I might go through this slide quickly because it's a bit maybe not as interesting, but just to say that we can ask now various parameters, how various parameters of the ecosystem affect this kind of question of, whether switching to organic is going to be beneficial for productivity. And so we see some various properties. For instance, a higher herbivory coefficient, RH, means this is going to be more useful to switch to organic, and that's simply because the reason switching to organic is useful is because it will kill herbivores. So if herbivore pressure is strong, it makes sense, and if it's not, the other way. And we can see some other sort of parameters where it's a bit less obvious a connection. For instance, if nutrient loss rates are higher, apparently going to organic is going to be more beneficial, which is, I would say, not as obvious at all, but this is at least what the model suggests. Yeah, so this is basically mostly it. I hope I didn't go through this too quickly. And so what I kind of, just to summarize things, I hope I kind of showed you the state of the green-brown food webs across different ecosystems and various kind of properties of that. Some sort of information from the current state of affairs suggests that there's sort of some coordination, if you will, between the green and brown pathways. That is, in some form, in some sense, the primary producers and herbivores that eat them are somewhat similar to the sort of detritus, the dead material, and the detritivores that consume them. And there's no, to my knowledge, an obvious reason why that should be the case. It certainly is a nice symmetry, which is kind of nice, but I don't know why that is. I can make some hypotheses, but I think to me, it's quite interesting to understand that in some large scales, if you will, these two parts of the ecosystem function very similarly, apparently. And finally, how do these kind of green brown food webs respond to nutrient enrichment? And for instance, in particular, there's some interesting differences between how the system responds to inorganic nutrient enrichment versus organic enrichment. And so for me, at least, one question is, well, what do I do now? Where do I take this stuff from here? And personally, I've always been interested in kind of the spatial aspects of ecosystems. So a natural sort of stepping point is say, okay, how does these kind of systems now work in a sort of in a spatial context? And a very interesting sort of system in that respect is kind of agricultural systems, and in particular, agricultural systems like, for instance, are quite common in Europe, for example, where you have this kind of patchwork, as you kind of shown here by this cartoon, of some forests, some crop fields, some other fields, and how do they kind of interact together in this sort of landscape scale. And what I would like to do is kind of connect this kind of complex landscapes like this together with this kind of ecology motivated meta ecosystem network stuff, which I don't know if you're familiar with, but this is a whole kind of paradigm in ecology of how to deal with spatial ecosystems. And basically how I have this kind of some ideas of how to kind of connect these two things by looking at the interface regions between different landscape, between different fields, basically. And more generally, some general thoughts I have, at least, and we can maybe discuss if that's of interest, is well, how can we confront data if collected across habitats? So if we're talking about spatial systems, how much data do we have about what happened when you cross habitats? If you have a field next to a sort of a more natural area, or two fields of different types, how does they impact each other? I think there's been some studies on this, but I'm not quite sure how to go about this, getting the data, and also from a modeling perspective. The question of parameterizing intensely managed ecosystems, how different is that from natural ecosystems? At the end of the day, I managed to collect a sort of what I think is enough data to get a rough idea of what happens in natural ecosystems. I think for some of the properties, I would guess it should be very easy to know this stuff about for managed ecosystems, because like anything else, especially when money is involved, people want to know. So people, I presume, there will be a lot of data on how plants, some crop plants, react to nutrients, for instance. But how predators and herbivores and things like that happen in the field, maybe much less so. And whether it really matters that they're somewhat different in natural systems, maybe it's about the same. One thing that occurred to me, I didn't get too much into this, but I quickly mentioned before, this effect that looking at food webs, at least, I could find things for aquatic food webs via this EcoPath system sort of software, but there's virtually nothing for terrestrial systems, which lead me to wonder if one could kind of make an EcoPath of sorts for soil ecology, looking at sort of fluxes and stocks in a kind of consistent way, looking at soil ecology. And in practice, the last question is, as I said, I kind of want to go into a landscape ecology like you kind of see here. And one question still that I have in my mind is, what's a good first step? And I have some thought on this, of course, but to me, that's still an open question. Yeah, that's it. Thank you.
Thanks, Yuval. Nice. Any questions from?
If I may ask.
Yeah, go ahead.
Yuval, thank you for your talk. It was very interesting, I must say. I never thought about all these things. Small question, in all your models, in many times when you look into a system, there's a limiting nutrient. So it's not that you add something and immediately the system responds as if there were no other limiting nutrients. So did you ever take that into account? Because it seemed to me that whenever you added something, there was no other limiting factor. It was just the system, all the system would go in one go just because you added one thing, while in reality, many times there is a limiting factor, which is different.
So just to make sure I understand you, are you asking, for instance, about what happens if there's two limiting factors like nitrogen and phosphorus, things like that? Is that what you mean? Or
I'm saying, for example, you add nitrogen, but there's phosphorus limiting in the system
right.
or something.
Yeah.
I mean, I can see the benefit of what you did by learning the shifts general, but taking it into the, I would say, real world. How do you say?
Yeah. So this is something we've actually had quite a lot of discussion recently, and also at the time when we started this project. And the short answer is that, well, I guess there's two main things I would mainly think about. One is that in practice, in most cases, when you have nutrient enrichment, you don't tend to have it from a single type alone. That is, in most, both natural and human impacted situations, you won't typically have a situation where you're only adding nitrogen. In practice, you'll add a bunch of stuff in effect. So in that sense, it's not likely that you're going to see, the response kind of due to a single one. And I totally agree that if I really, I'm here, and from the model perspective, I really assume it's only nitrogen limited, which is, for instance, for terrestrial systems, that might be more the case, and sometimes for aquatic systems, that's more problematic. They're often more phosphorus limited in effect. But in my mind, the main point here is still qualitative in the sense of the modeling. It is really just asking, well, essentially the kind of sensitivity. How are the different compartments more sensitive to these sort of nutrient enrichment? And so, as long as you don't assume, that the thing is entirely not limited by nitrogen, for instance, and something of what I've shown here should be seen. And so this combination of the fact that, in my mind at least, the focus is more on sensitivity, and on the other hand, in practice, the additions tend to not be only nitrogen, means that I think it's reasonable to take this kind of result as a guiding kind of line. Certainly, it would be very useful and interesting, to be sure, to go at it more properly, to look at stoichiometric aspect and so on. I would just mention that it's interesting. I find that, it makes a lot of sense. But for some people, the first thing that would come to mind is these kind of issues of stoichiometric and different limitations of nutrients. And for some other people, the way I model the food web would be like a big thing, right? And all of these things are quite legitimate. There's a lot of assumptions that are put in. But if I'm being kind of realistic, then, when I kind of estimated the stocks and therefore also kind of influxes, and that's, I think, reasonably done. As in, I feel quite confident about the reasonable estimations there. When it comes to properly the modeling predictions, I would only take it as a qualitative level because there is so much unknowns. I didn't go into that. There's a lot of unknowns about the levels of things like self-regulation of the different compartments, for instance. As you said, there's a lot of unknowns about the nutrient dynamics, but as I understand it from one of my colleagues who's more into nutrient dynamics, it gets more complicated than this even, because it's often unclear how different aspects of the nutrient limitation play out. And so there's a whole bunch of unknowns, and I guess my feeling is that this is the best that can be done at this time. As I kind of effect, I would say that at least as part of the data collection, I really honestly assumed 2 years ago that the data would just be there, and it just turns out that it wasn't. And so to me, just an initial step of saying, oh, look, this is the state of the system, and this is kind of the initial reaction we would expect from nutrient enrichment. To me, that's the kind of the way I would view these results. And I'm sorry, and one more point is that indeed we also wanted to be able to compare the different ecosystem types to some degree. And so that's kind of also, you have to make some simplifying assumptions there as well. Like if you want to compare between a forest and a lake, obviously these are very different ecosystems. So just to be able to kind of get some handle on it, it helps to make some simplifying assumptions.
Yuval, I have a question. What about environmental conditions above the soil? Do you include it in the model? What happens if climate conditions are changing?
Yeah, I don't explicitly do it. It is done in effect. Well, here, if we look at the model, this is the model with some 20 parameters, for instance. And so just as an example, some of the things of the, for instance, of the properties of the food web, the plants, herbivores, insectivores, and so on, they have their consumption coefficients and mortality rates, and some other things as well, but mainly these ones. And so, for instance, it is reasonably known how mortality rates are changing by temperature, as an example, and consumption coefficients not too badly. Some other things are not as clear, so it's just self-regulation, but nonetheless. And as well, mineralization rates, nutrient loss, and things like that. You can try to look at those, indirectly, how those are affected by temperature. And so I have, for instance, worked on a project where some of these sort of food web properties we looked at, and for changing temperatures. I could imagine trying to do that in a more larger scale, as in kind of include all of that and try to parameterize all of these things. My guess again is that we would end up with a very, very rough prediction that I would put it this way, right? We did something for a much simple system with kind of basically, two compartments of resource and consumer, like plants and herbivores or something. And already there's quite a lot of unknown. I don't know well enough how, I would actually assume that the sort of the substrate dynamics, the nutrients and stuff, I would assume that those are better understood from a temperature and other condition dependent. Although I don't know this, but I would just kind of guess. So maybe that part wouldn't be as bad, but nonetheless, if you try to add up all these things, I think you could definitely, if you kind of pose it well, you could probably find some interesting things that are probably hopefully somewhat robust. But let's say the distance from there to going into kind of reasonable, like quantitative predictions that I would trust would be quite a distance.
Thanks.
William, may I ask a question, please?
Yeah.
Okay. I just wonder, I'm looking at the model, and I actually wanted to see it in the beginning. So how do we know that all these effects are linear by variables? So why it just becomes first order equations? Why there is no square or more complicated equations? How do we know that?
Oh, well, the quick answer is that we don't. This is basically a linear assumption. So the first order, the simplest modeling you can do. And so there's two things here, basically. One is that this setup is simple, and that means that, for instance, the model is also used for some of the estimations to kind of do them indirectly, because there's some things that are not known directly. And in that sense, it's just a simple way to do it. And it shouldn't really affect that aspect. Though that's debatable, I suppose, but to a large extent, I think that's reasonable. The trouble really comes again when you make predictions with the kind of the model dynamics. And I mentioned this briefly before. This is certainly a potential limitation. This is what is called a type 1 response in ecology. These kind of sort of linear-like relationships. Certainly for some of these compartments, especially these kind of terms here, you'd usually kind of expect sort of a curve that goes down. So for instance, as you start having more and more nutrients, they're going to be less and less useful for the plant, certainly, and so for some other compartments. So we actually tested it to some extent, and it doesn't really impact things too strongly under reasonable assumptions, let's say. That is, we don't know. The main point is that we don't actually know at the ecosystem level at all how to model that. Like it's virtually unknown. There's some specific systems it's understood for, but usually at the sort of species level. So for instance, how do rabbits eat grass? Maybe in that level you could understand that, but if you're now asking how do all the herbivores eat all the plants in a grassland system, that is generally not understood at all. And so we'd have to make assumptions anyway. And so under reasonable assumptions, the predictions don't change much, certainly at the qualitative level. But you're right, it is a limitation, and like many other things, to me, this is another. Yes, we really, to my knowledge, I don't know. We don't know how to do this.
Okay. And
Yeah.
one more question is, maybe you even asked it when Shahar asked it. So I reminded sometimes there are kind of the threshold effects. I never touched that field, but I had seen some discussions and basically may obtain not only nonlinear effects, non-exponential, but really like sigmoidal shape. Or there's a threshold effect of food, of of nutrients, whatever. So, which means probably it happens when you have control by transport or supply, whatever. So, is it accounted somehow inside of the model?
I'm not sure. I don't think I quite understood you, but I mean,
Okay.
actually, the main question is about threshold effects, which I don't really know what you mean with that term. But I would say this, and it might. I hope this kind of this might answer. So, to a large extent, if you look at most of the terms here, you have this kind of this is a consumption term, and or rather two consumption terms. You consume something from below, and you consume from above. And then you have this kind of just constant mortality rate. But what is perhaps quite important is this term here at the end, which is self-regulation. And so what this self-regulation effect does, and this is according to some recent papers on this, is that to a large extent, it controls how much a compartment is, if you will, limited from below or limited from above. Effect. And so what I mean by that is if self-regulation is quite strong, for instance, for plants, then plants don't really care so much about the herbivores above them. But if it's quite low, then typically they would care quite a lot about the herbivores above them. And so this is kind of a relatively simple way to sort of switch between those two things. Having said that, and I do hope this is actually going the right direction of what you were asking, estimation of self-regulation is, again, non-existent. Maybe for plants to some extent, but again, I don't think so much at the ecosystem level. What we ended up doing is that we had some reasonable assumptions for them. And it seemed that most of our kind of main conclusions don't change by these different assumptions. But I mean, I can't say that I feel certain that that a better estimation of these self-regulation terms will not impact things. But I would just, to clarify that self-regulation effect, for instance, would say, so for instance, part of what we saw is that, for instance, if you put in more organic nutrients, it will kind of feed the brown channel and end up allowing plants to grow more potentially. That kind of stuff is quite impacted by self-regulation. And so it is possible that if our self-regulation estimations are way off, that kind of prediction might change. But I think we have a reasonable handle on those things. I do hope that was kind of the right direction. I'm not quite sure.
Thank you, Yuval. Thank you.
We have some more time for questions if anybody has. So if not, then I would like to thank you, Yuval. And thanks. And we will have, I guess we will see you next week. Not you, Yuval, at the next seminar. Thank you, everyone.
Thank you.
תודה רבה.
בבקשה, בשמחה.